Hello, everyone. Hopefully you can hear me. My name is Jesse Bloom, so after that very sobering video, I'm going to be leading this first session, which is Biorisks in the Future. So we're fortunate to have 4 excellent scientists on our panel today. I'm going to briefly introduce them and refer you to the online material for detailed biography. We have Volker Thiel, who's at University of Bern, Gustavo Palacios from Mount Sinai, Clare from University College London, and Ravi Gupta from University of Cambridge. And our goal on this panel is to discuss some of the scientific issues and sources of risk for sort of biological events in the future. So the first question, which I'll start off leading with Volker, is sort of what do you perceive as the biggest risks that we face going forward, both from zoonotic and biosafety, biosecurity perspectives?
So I think you already distinguished in your question between zoonosis and the other aspect is biosafety. Let's start with zoonosis. I think it's quite obvious that we essentially invade nature at the moment. We have much more contact to wildlife, and I think that's where there is considerable risk. Also the way how we deal with, let's say, animals mainly. You may know that the markets in China have been one place where at least it can be considered that virus can spread easily. So our behavior, how we deal with nature, how we deal with animals, how we live essentially, our lifestyle even, is maybe concerning zoonosis one of the most important way how we can maybe generate
and I think that's a good point. I mean, I think when we think of the markets, we think not only of wet markets like we discussed in China, but for instance, we saw an H5N1 outbreak on a mink farm in Europe, and I think there's lots of things like that are generating risks.
So the other thing when it comes to biosafety, biosecurity, let me maybe start with where I have not a big feeling that there is a big risk. I mean, risk is always the question, if something happens, what impact does it have and how likely it is to happen? And I think in many countries we have very good regulations on the work that is done with, let's say, these high pathogenic viruses or pathogens. These are countries, let's say, where people stick to the law, where you have regulations, where everything is also on the technical standard that you can be quite safe. It does not mean that nothing can happen, but I think here we are rather good. Where I have a little bit of fear or where I think there are issues is in areas where it's not regulated properly, where we maybe have research outside any regulation. So this is sort of maybe the candidate areas where we should do something.
Yeah. And what would you see as examples of that sort of research that's going on in areas that are outside of the standard regulation?
Well, those examples you can hardly name because they don't tell you I'm doing something outside of regulations, but you can imagine that those things can happen.
Yeah, no, those are very thoughtful answers. And I'd like to also pass that question to Gustavo, and maybe, Gustavo, you could also elaborate a little, as people may or may not know. Gustavo's now sort of an academic scientist like all of us here, but prior to that, you were working at US AMRID, which is a different sort of more regulated or highly regulated environment doing research. I'm curious if that gives you any perspective on these issues.
Yes, I actually am going to mention and follow up with the word, one of the words that is regulations. The other word that David Relman used in his introduction was the strategic ambiguity and the fact of how, in terms of our analysis of the risk, we are considering always that there is a careful evaluation of the risk before we are taking an action. And it seems that one of the major risks that I am seeing now is that we have a very nonchalant approach to do research and on high containment pathogens or potential high containment pathogens. And so in the past, one of the ways to handle that was to ensure that national laboratories and government laboratories were involved in a lot of those types of research. And those are heavily regulated, slow, very bureaucratic, but there is a reason why they are like that. We have moved a lot of that research to academia that actually is a lot less regulated. Maybe that is a problem that we need to consider. And the second aspect is, at least in the US, government scientists are not as prestigious positions as they have been in the past. And therefore there is a degradation of talent in some of those areas that maybe are not attracting, the pool of individuals that could sustain that type of work in a very regulated way and advancing the science that is needed to control these risks.
Yeah, no, that's a good point. And I think Gustavo makes a valuable point here in terms of a lot of research in academia, not having the same amount of vetting and review and regulation as research in some sort of government labs. On the flip side, and maybe as we go to Clare and Ravi, I mean, we have seen during the COVID pandemic, there've been a lot of academic scientists, both of you being examples, myself being another example, who because of sort of the flexibility and relatively lighter regulation in academia were able to quickly pivot from working in other areas to working on viruses like COVID-19. And so I'd also be curious to hear your perspectives on what you see as sort of these risks and, both from zoonotic and biosafety perspectives.
Thanks, Jesse. I think UCL would argue that we weren't more slack on our safety, which allowed us to work on SARS-CoV-2. I think the guys summed up a lot of the same points that I would have made. I guess there's two, in terms of zoonosis, absolutely increased interaction with animals, and I think that obviously involves intensive farming and wildlife trade, but also I think something that's quite important is obviously climate change. So increasing not only the frequency of our interactions with animals, but the types of interactions we have, particularly changes in vector distribution. So mosquitoes, ticks, for example, we're seeing in Europe increase in frequency, and tick and mosquito-borne infections that we haven't had in the past because of changes in ecology. So I think that is an important contributor to increasing zoonotic risk. In terms of sort of biosafety and biosecurity, I agree very much that in a lot of the world, particularly the UK and where most of us are working, everything is very heavily regulated, tightly controlled. Safety is paramount. I guess something that I've been thinking about over the past couple of years is field sampling. So people going into taking ecological samples or samples from animals, either intentionally seeking to understand what potential viruses and bacteria may be there, but additionally people sampling for other purposes, such as routine surveillance, or groups who may not be thinking about what might be in the samples that they're taking because it's not what they work on. They're not virologists, for example. So how are those samples taken? How are they stored? How are they processed and worked with for people who are not maybe as tuned into thinking about the risks of what you're not used to thinking about? So I think that, for me, is something that is an important consideration.
Yeah, I'd like to add, I think, to that the fact that unintentional exposure is probably quite a big risk. For example, if you're not working on a particular virus, but you happen to have a sample that has a different virus in it, this is, I think, a really important area where frameworks that are adopted globally are important. We do need international frameworks for processing animal samples, whether it's blood, tissue, or other products. So there's certainly something that can be done there, because it's fairly. But then the question will be, of course, if you identify a framework, which labs have the capacity to process samples within category 3 containment, for example? Or do you derogate everything to category 2? So some of these will be pragmatic decisions, because it's going to be quite risky to generate a framework that very few people can adhere to, and that actually polarizes the world into those who have and those who don't have. So I think that is tricky, but absolutely essential. Just going to the question about zoonosis, I think that, yes, it's a huge problem. I can't see it getting any better through regulation. I mean, of course, many of these things are entrenched in society. We are, of course, becoming more intensive in the way we do everything. It's very hard to see how we will draw back from, man's quest to, to generate capital and wealth. So I think that the way to mitigate that, it's ambitious, but I think it's something that maybe the US government is already thinking about doing, which is to categorize and document all, as many different varieties or viruses that are out there, and then generate, ranges of vaccines, for example, so that you have an array of vaccines against different groups of viruses, so that if there is a spillover event and we manage to detect it, that we can rapidly scale up vaccines, for example. So you could look at it from a mitigation point of view rather than how do we stop the source of that spillover, which I think is a much bigger, well, they're both equally large tasks, and maybe what we need to do is a bit of both.
Yeah, that's great. And to kind of play off that, because Ravi's sort of leading into my next question, which is sort of what can we do to mitigate these risks? Yeah. So Claire, you and Ravi were both sort of highlighting, that potentially there could be risks of collecting, unknown samples to study. And one question I'm interested in is what you see as the lessons that we can draw from what actually happened with the COVID-19 pandemic for research going forward. So for instance, when we look at the COVID-19 pandemic, despite a lot of interest in, trying to identify potential zoonotic viruses ahead of time, COVID-19 wasn't identified until it started to spread among humans. We didn't succeed in identifying it ahead of time. So you could argue that was a failure of the scientific approach we were taking. On the other hand, I remember back to early 2020 when you heard people say things like it will be 3 years or 5 years until we have a COVID-19 vaccine, and in reality it was really only about a year until we had a vaccine rolled out that was starting to be distributed around the world. And I guess in general, one of my questions for each of you is when you sort of look back over what's happened with this pandemic, now that we've actually lived through one of these types of events that we're all trying to work to mitigate, what are the lessons you draw about how we should be thinking about what to do going forward?
Well, I think you could spend hours in discussing that. I want maybe to take two examples. One is teaching and outreach. I think we live in a world of polarization now. I want to bring one example from my university, where we have a nice course that's called the Host Pathogen Colloquium, where we pick papers and discuss with students. And I usually pick papers that are controversial, that touch the topics of biosafety, biosecurity, and things like that, gain of function work. And it's a little bit surprising to see that the students don't know that there are any regulations. So there is also, in the public opinion, people think these scientists could do what they want, and it's not like that. So I think that's something where we have to reach out and tell people how it is and what type of regulations we have. And then, of course, we have to discuss, is this sufficient or not?
Mm-hmm. Yeah, that's a good point. And I do think it kind of emphasizes that forums like this that are bringing these sorts of exchanges are, in my experience, pretty rare. Gustavo?
Yeah, I guess that my point is we know how to do the work to describe the virus or to describe viruses that are near the jump or viruses that have spilled over already. We know how to assess the risk of each of those activities and what we need to do to reduce the risk to zero. It could be reduced to zero. What we don't know, actually, is that we are not putting enough pressure in ensuring that the training for the people and the acknowledgement for the people that are doing those experiments or those expeditions or those field trips I mean, we know what type of PPE you need to do to go to a cave and collect samples with very low risk. But if we don't follow those, if we approach that with low level of training, that's where we are failing, is enforcing our knowledge. Because we already know we have generated biosafety laboratories at level 3 or level 4 that could handle that risk. We should be able to understand that if what we are doing is describing the virome, we don't need to collect the live sample. We can inactivate the sample at the moment and there is zero risk because it's completely inactivated and we are only recognizing the nucleic acid in that sample. I mean, we know how to deal with the risk. What we are lacking is in professionalism.
And so what, I mean, I know Volker was talking about this course he teaches. What would you see as the things that could, you know, what do you see, Gustavo, as the things that are needed in terms of training of the scientific community or of researchers on these issues?
So, I mean, to make some examples, we know it's not under dispute that some events in that work that should have been done at the higher level of biosafety was conducted or was being conducted at BSL-2 conditions. That is not disputed. I don't know, I want to take, so one of the comments was, well, that you made was actually that the information that was collected before the pandemic was not useful to control COVID-19. Well, that is our fault. I mean, RAG-13, even when it's different at the 96, 97% level compared with SARS-CoV-2, could have been very well used as a method to explore how to make a vaccine, and probably a vaccine that could have the spike against these viruses could have been at least grossly protective against SARS-CoV-2. We could have, I mean, we knew that clade of viruses was there and was circulating. We just did not take any action to use that information to prepare. But the information was there. Sorry.
Yeah. And Clare and Ravi, what would you say to those questions?
It goes back to the comments I was making about, you know, having an approach which does high-level surveillance, intensive surveillance, and then acts upon it in a concerted fashion. But that will take, you know, a large amount of cooperation and funds to do. But that certainly is something that I think we should be advocating for, because that will be the ultimate application of science, really, to generate an array of vaccines, even antiviral compounds, against an array of viruses. So I think that's something positive we can at least advocate for as a community.
Yeah, I think it's very hard to predict, though, what, you know, the next pandemic will be. I guess in hindsight, everyone would say, well, we should have predicted SARS-CoV-2 in a way because coronaviruses are particularly good at jumping species. They've done it before. Influenza viruses are very good. So I guess more broadly, I think there were a lot of general positives that came out of what I think I've learned, and, you know, hopefully my colleagues have learned as well, about you know, engagement, data sharing, openness, transparency. You know, I think we saw the best of science in how scientists collaborated and openly communicated in the response. So I think, you know, trying to identify what are the likely potential pathogens is worthwhile, but having processes like we do now in place to respond even while we're trying to figure out the biology. You know, I think we did a pretty good job at lockdown, engagement, responses, social distancing, all of these sort of containment measures that got, you know, the R number down, that bought us time to understand the biology of the virus, to get a vaccine and to start to, you know, have some therapeutics. So I think that kind of framework is, you know, how we respond to a respiratory pandemic, how we might respond to a pandemic of, say, Ebola-type virus or Mpox. You know, we can have different frameworks in place for how we would respond to that if it was something unexpected.
Yeah, and Ravi?
I'll just follow up. Yeah, I mean, it's interesting. We got away with it for SARS. I mean, obviously, millions. You know, we still lost a lot of people, but the vaccine, as you say, took something like a year to bring to market. But we used lockdowns, for example, and other methods to slow things down. Now, the next pandemic, you know, we're already talking about whether we should have done a lockdown, right? So the next time around, the public won't accept a lockdown, let's say. So we won't have as much time to get these vaccines, you know, together before before large numbers of people die. So I think that's also a thing to consider.
Yeah, so and certainly public health responses is a very difficult question. I guess I want to return quickly to thinking about the vaccine. So thinking back to what David Relman was talking about in his remarks at lunch before we started this, is he was talking about how now, due to these new capabilities, sort of many researchers can take the sequence of a virus and just create the actual physical instantiation of that virus using reverse genetics. And he was talking about some of the potential attendant risks there. On the flip side, I would point out with mRNA vaccine technologies, we saw both academic researchers and probably more importantly, companies like Moderna and Pfizer having that same ability to simply take the sequence of this spike protein and essentially have a template vaccine in a matter of weeks, whereas, you know, in the old days, which were not that long ago, it took a long time to produce a vaccine. And I really think it's interesting to think about this as sort of two sides of the same coin. Many of these technologies that David was talking about, the ability to do rapid gene synthesis, to identify and sequence these things, also gives us the capacity to make these vaccines in the same way it gives someone the capacity to make the actual viruses. And I guess, do any of you have viewpoints in how the thinking about those new capabilities should sort of change how we think about preparation for both zoonoses and biorisks?
Well, I mean, it is all related with transparency. I mean, the information, but the quickness that you get information that you can use depend on that information to be available to everyone or publicly available to people that actually could act in generating a vaccine. So we need a new system to encourage that transparency. We still, for COVID-19, we still do not have full transparency from all of the actors. So that obviously it's a huge problem that essentially if you keep for yourself certain information, I mean, then all the good things that could come from the action from that technology could not be usable. Now, other fields, and I like the idea of the Bulletin of Atomic Scientists to take the lead on this, is because in the nuclear aspects, we have deal with these type of problems. There is an international agency that essentially handles and supervises and look at what all the states are doing, ensuring that there is openness, ensuring that there is transparency, ensuring that when a state do not help, I mean, it's a non-cooperative state that could actually be sanctioned by the international community when it's not open and transparent. That needs to be that should be now the norm probably to other areas, like in the area of biosecurity.
And just to sort of elaborate on that analogy that you're making there, Gustavo, I mean, I think that the Bulletin of Atomic Scientists also provides a model in terms of scientists thinking about the responsibility of this. But on the other hand, when I think about many of these capabilities, both the capability to make at least a preclinical vaccine or the ability to generate a virus, it's not like producing a nuclear weapon, right? There's multiple people just sitting up here on the stage who would be able to take the sequence of a new coronavirus or new influenza virus and both produce that virus in their lab or produce a vaccine candidate against that virus in their lab. So it's sort of a highly decentralized capability, which I think we've seen many of the ways that it can be used for benefit, but you also don't have have to have a lot of imagination to think of some of the ways that it could be used to detriment. And so how would you think about that if this is not something like nuclear technology where it really requires highly specialized, high-speed centrifuges and uranium, but it requires, advanced biotechnology, but the type of biotechnology that all of us have in our labs, as do thousands of other scientists around the world?
And maybe I can comment on that. So first of all, let's say you generate a virus from a sequence that is essentially reflecting the wild-type virus. So I don't see a difference between this copy and the wild-type virus in terms of biosafety. So the same phenotype is expected, and you treat it with the same safety measures. So what we discussing a lot is this so-called gain of function, where everybody has a little bit of a different view where it starts and where it ends. And here, anyway, the scientists are responsible to tell the authorities what they are doing, what risk they think this may have, and then it's weighted against benefits. And we have to come, and that's also the purpose of this meeting, I think, to come to some ideas how we evaluate that, where is there any natural border that we should not cross? And that's maybe a constant thing. It's maybe not even what we think today may not apply tomorrow. We may shift our view or change our views where the borders are and what we should do. And the other thing is what people often think is that you have a sequence, you have a virus, you design something, and then you have the desired phenotype. It's not like that. We don't know enough. Whenever you study those functions of a virus by reverse genetics, you're surprised how you can mess up things. So in terms of the virus doesn't grow anymore, it just is not viable. It is creating something new is, with our knowledge, barely possible. So the people probably think we are better than we are.
Yeah, that makes a lot of sense. I agree with that.
Yeah, that's a really good point. I think any of us who work on viruses know it's much easier to break the virus and kill it so it doesn't do what you want anymore, it doesn't replicate, than it is to give it a new or enhanced function. And I think you're right that the conversation absolutely needs to be had, but gain of function is not one particular area of virology. And what is your gain of function in antibody escape, for example? You may view that as gain of function. That's incredibly important to know how a virus might evolve to escape a current vaccine or might evolve to escape a drug. So anything that changes the biological properties is not necessarily gain of function, and that gain of function research is not necessarily a bad thing. And I think that's a really important conversation that we need to be having with regulators, funders, the public, and I think this is a great forum to start that conversation, because I think it's become a very loaded phrase without people really understanding what it means, and a one-size-fits-all regulation is not going to work in this regard, I don't think.
And just to add to that, yeah, I agree with all that. So just to add, the layer of sort of oversight at university level, of course, varies between institutions, and this is quite a big burden, of course, for a committee, as David was mentioning earlier, to have a proposal, and you're having to weigh up the pros and cons, and potentially the level of expertise or experience on those committees will vary, and some will be conservative and then say no to things, and some you can imagine being more liberal. So I think that is a problem, and so that's why I think actual, so explicit guidelines are really quite important that could be circulated.
And to sort of get to that more specific question and allude to what a number of you were saying, because I completely agree that this term, gain of function, is an extremely broad term that very many things, perhaps a small number concerning, a very large number probably not concerning, fall under that umbrella. How would each of you think when you think about your own research and how to draw these lines when you're working with different viruses? And as I ask that question, I'd even specifically call out 3 different types of viruses, broadly speaking, that you could imagine working with. One is a virus like SARS-CoV-2, which is widespread in humans. Another is a virus, like we were discussing in these virus discovery and looking for the next potential virus, which is a virus which hasn't infected humans, but who knows, maybe it could. And then the third category might be something like the 1918 or the H2N2 influenza, which is a virus that we know can infect humans, but isn't in humans right now. And I guess I'm curious how each of you would think about, in your own research, how you would kind of put these cost-benefit analyses when you think about that type of research.
So the rule that I have been following, and actually that I take from the risk assessments done at USAMRIT for Dietrich, is that you only should work with something that is currently circulating in nature. If it is not currently circulating in nature, there is no need or reason to work with that pathogen. If we are changing something on a virus that you have not observed in nature, a change in an area, you're generating essentially a chimera that will need a different level of risk assessment that probably should not be in the hands of a scientist to decide. It should be at a higher level, or where you don't only have scientists, but you also have regulators, biosafety experts, and biosecurity experts. And I think that that is a reasonable, I thought that that was the rule everywhere. And again, I go back to that nonchalant state of assessing the risks that I mentioned at the beginning. I think that that is the major problem. If we are not aware of the risks, there is no way that you can minimize them.
Any other thoughts on this topic?
I mean, I would agree. I think making something, working on something that exists in nature, or modifying a virus in a way that reflects a natural circulating COV variant, for example, because you want to understand function of a gene. Yeah, I think mixing spikes, creating a whole new virus with altered tropism is very risky. And I think people need to, there's experiments you can do, and then you need to sometimes reflect and think, is there, something you and I were discussing yesterday, Jesse, is there a better way to do that experiment? Here's the first option to do that experiment, which is modifying the virus. But what if I modified the cell, for example? Is that a safer way to get to the same answer? And I suppose, you could think of it from the worst case scenario. If something went wrong, can I justify having done that experiment? Can I justify having made that viral mutant? So kind of, I think you need to just pause, but just because you can do the experiment doesn't always mean you should, and reflect on, if something was to happen, am I comfortable that I could defend the work that I was doing was absolutely necessary?
I mean, to add on that, you said changing, for instance, the spike in the coronavirus. Also here, the borders are not clear. For many of the variants, we're talking of a handful of bases that are different.
Oh yeah, I meant something more like putting MERS spike, completely different virus. Yeah, absolutely.
Exactly.
Yeah,
so also here we have to
that's
consider.
Where it's very grey, isn't it? Yeah.
Exactly. Yeah.
And so I hear in some ways Volker and Clare coming down on this criteria, which has actually often been used in the regulations. Are you making something that could reasonably expect it to occur naturally? Is that almost what you're suggesting? It sounds like. Ravi, you want to?
Another way to of course approach this is to have reverse genetics as your last set of experiments, and you start with pseudotypes, for example. So you don't have to go straight to a live virus to demonstrate or to explore the scientific basis for genetic changes. You can just start with simpler systems, virus systems, using the spike protein on a lentivirus, for example, and just manipulating that, which is completely safe. And you can find out 80% of your facts that way, and then you can make the live virus if you can justify it.
Yeah, and I do think that Ravi makes a very good point there in the use of, for instance, in studying coronaviruses, you can make pseudoviruses which don't pose a hazard to humans. And we've also seen, many examples where researchers have taken these same technologies we're talking about and, for instance, created even replicating coronaviruses, but where they've made intentional modifications that will attenuate those viruses, such as removing key accessory proteins. And so I do agree that perhaps in many cases there's ways that we can think intelligently about using the technology to actually make these experiments safer. I don't know if you want to elaborate on this. I know we were talking a minute ago about work that you were doing.
That's a classical example of if you want to study a function of, let's say, a viral gene, you would knock it out, for instance, and you would expect loss of function here. Like you said, a virus that has lost one function. And here I don't see any critical points.
Mm-hmm. Yeah. So to go to this question where we can, think about sort of in what are many cases these gray zone judgments, like is this a virus that would occur naturally? Is this risky? Is there a safer way to do this experiment? I guess the last big question I have for all of you, because it's a question that I wrestle with a lot, is sort of how do we have these discussions? There's a lot of controversy around this. As you know, there's a lot of people in sort of our own field of virology that sort of feel like we're under attack as being irresponsible. There's a lot of people out there in the public that feel like virologists aren't thinking carefully and are being irresponsible. And it's become, in some ways, a very non-productive and conflict-prone discussion. And what do you think are the things that we can do and can encourage to sort of improve the quality of that dialogue and recognize, how we being both virologists and people worried about biosafety, work together to sort of get to a better place?
I mean, I think that's probably one of the most important sort of questions there is, in terms of how do we move on from here? Because clearly basic science and virology has been immensely important in human health with pandemics, throughout the last 50 years or more. So we need to continue this type of work because if it doesn't happen, of course, we'll be blamed for not doing it down the line, number one, of course. And there is, a moral imperative in a way. We have the tools to improve human health and prevent disastrous pandemics from killing people. So I think it's a process of rebuilding trust, number one. Communication, of course, but this is really hard now, of course, with social media. To get the truth out now is much harder than it used to be, probably, because there are so many voices from different directions. So again, a coordinated effort it's going to have to be a supranational organization, I think. It's going to require money, but I think investment is going to be critical because, of course, you're looking at public trust in science at the end of the day. And I think it's in government's interest to promote that. So concerted sort of effort to rebuild trust in the scientific process is important. We can remind people about vaccines and how, and actually the benefits they've brought. Of course, there are counter arguments from people about, side effects. So there are a number of issues that need to be kind of taken one by one. And the facts kind of replayed to the public because, of course, we're now in a situation where we're hearing stories about why we shouldn't have done lockdown or why we shouldn't have closed schools. There's a lot of that revisionism going on because people have forgotten about the things we went through. So I think that that this is a really important time for the world's public to be reminded of what science did during this period. And then once you've got there, then we have to say, well, we'd like to stop this happening again. And of course, everybody will buy into that. Because I don't think some people did benefit from COVID-19, but most people didn't. But I think there's a period of rebuilding trust that's essential.
Yeah, I would second that. I think somehow trust has been lost, and I think social media and misinformation is not helpful there. So I think we have a responsibility to engage and educate. And I think Ravi made a very good point about it being a collaborative effort. So I think this type of meeting is a fantastic beginning. So we have responsibility as scientists to behave responsibly. Journalists have responsibility to report responsibly. And then above that, you have institutional and governmental responsibility, but something as well that we could think about is, a responsibility or an interaction with funders and reviewers of grants and reviewers of papers. I was talking to someone the other day about how when you just kind of making sure that people are aware of there's a responsibility at every level to reflect and audit and make sure that things are done safely and promote, you know, the positives of science. So it definitely has to be a collective effort.
Any thoughts, Gustavo or Volker?
I feel it's a difficult point for me to try to explain. I am a virologist and I am a scientist, but for the last 10 years I have been working in government in a biocontainment laboratory and handling risks and biorisks. So I have seen the other side of biosafety and biosecurity with a heavy impact on what I have been doing. One of the feelings that I have about what's happening in all of these discussions to scientists is that as scientists, we are not listening. The community is responding in a way that we are the expert, trust us. But we have not engaged with other parts of the communities during this whole process, where we accept that there are other points of view. The reconstructions of the majority of the discussions on the origins of COVID-19 demonstrate the absence of experts in biosafety and biosecurity and biodefense in all of those discussions. We were setting policies or discussing how we were going to deal with the more likelihood scenarios, but we were not involving other people. Most of the discussion was among virologists and evolutionary biologists. So that is not listening, and that's why we are in the situation that we have lost trust from the public.
Any other thoughts?
I can echo what has been said, but maybe something more towards the future. I mean, things are always seen in a certain way at the time you look at it. So now, as was mentioned, people may not consider that a lockdown should be done anymore. And at the time we had the lockdown, people thought, oh, I want to be safe, I stay home. So we have to be clear that now we are coming out of a pandemic, and we see things maybe different now than we will see it in a few years down the road. So this process that we are starting here, or that we are having here, we have to revisit that. So we have to think about where are our borders, how should our regulations be, but we should not be too impressed now from the pandemic. Next pandemic might be something completely different. We may not get a vaccine because a simple vaccine like mRNA is not working. You know, there are certain things that we have to think about beyond this current pandemic, and have to consider that things are maybe different in a few years from now.
Yeah, I think that's a very good point. I don't know if anybody's ever read the report on the 1976 swine flu scare, which was a pandemic that never happened. But one of the points in that report that's always stuck with me is one of the people wrote, you know, pandemics only happen every few decades, which mean that scientists get to make very few observations and have a lot of time to think about each observation, and that's a circumstance ripe for overinterpretation.
But we should think about it and not forget, because people also forget in a few years from now. I mean, you know, you will see that funding is going down. People are not interested anymore. So we should keep that up and discuss.
Yeah, so I agree. And to sort of, I think these are all good points, and to sort of move on to Gustavo's point that part of our job is to try to explain our perspectives, and part of our job is to also try to listen to other perspectives. For the last 15 minutes or so, we wanted to kind of open the floor for questions, if there are questions. I guess I'll just call on people if there's not another system. Natasha, go ahead.
I'm taking my question. Could you tell me what you think the risks and benefits of the global Virology project are, and whether it's worthwhile to pursue it?
Okay, so Natasha's question was specifically, and it relates to some of the topics that were being discussed here, the Global Virome Project, which is a large-scale project to sort of catalog the diversity of viruses that are out there in nature perhaps with the eye towards identifying the next virus that might cause a pandemic. Does anybody on the panel have thoughts on this?
Well, I mean, it's an essential project, right? We have the technology to do it, and many of those viruses may have human benefit. So, you know, it's critical that we do understand what's out there, but in a safe way. So, as we mentioned, you can, if you're just going to look for sequence, you can inactivate the samples and just have nucleic acid, and there's no infection risk. So, if that's the aim of the project, then great, because you could always then resynthesize or look at other ways of examining those viruses that you're interested in. At the moment, you're going to try and catalog 10 billion new viruses or whatever, and that's purely a sequencing exercise. So, for me, there's no risk from the Virome project.
Maybe also commenting on that. I mean, we have maybe more risk in things we already know, and this was mentioned before. We knew that a SARS-like virus can emerge in people. We knew where it comes from. We knew from projects that sampled in bats that there is a high diversity of those viruses out there. So we knew all that, and I think it's to mitigate risk is better to then act and say, okay, we know that they are all there, so we want to prevent that they are transmitted from animal to human.
What's that? Oh, take a bunch of questions? Okay.
You know, the word trust has been mentioned several times, and it has many dimensions to it. One of it is the understanding of the scientific process. You know, scientists sitting in their ivory towers never really explain how the scientific process works. It works on the basis of data and how, you know, data has to be transparent, and that data to transparency builds trust. So, you know, the fact that WHO says masks work and then say masks don't work, the public has this perception that when a scientist says something, they're speaking the truth. But that truth also depends on the technology available. That truth also depends on many other circumstances. I think understanding the scientific process is so important, and this is something that each one of us, scientist or not, has a responsibility towards.
Yeah, I think you're absolutely right. Trust and education go hand in hand, don't they?
Hi. Following the discussion about trust of the population, we have talked a lot about transparency from researchers, from academia and governments. However, should we also ask transparency from the big companies? And sometimes, when can we draw the line between knowledge that have to be shared having a public health implication? What do I mean is that in this pandemic, we have seen that quickly academics have shared sequencings that then were allowed to do PCRs and have a rapid response. However, this same knowledge helped to have quick vaccines. But once we have the vaccines that work well, they're all on profit problems. And this is an issue that maybe is also we need to discuss, because it goes trust down, especially of inequality, of access, of all the response during the pandemics, seeing access for diagnosis as well as access of vaccines at treatment.
Hi, Tim Trevan at the back here with Chrome Biorisk Management. Not questions, but two quick comments. First, on regulation. Part of the problem with biology in general, not just virology, but biology in general, is the field is moving so fast that I don't think regulation is the solution here. It may be a component of the solution, but it cannot be the solution, simply because the regulators don't understand the subject to regulate it before it happens. So, second of all, if we regulate it, and in Germany may well have very good regulations, but other countries don't. If you regulate in one part of the world, water flows where it flows. The science will go to where it's possible rather than where it's regulated. So regulation And engineering also doesn't really work. Some of the biggest incidents we've had in the field of biosafety and biosecurity risk have been in the most competent, most funded, most engineeringly advanced facilities. We have to take into account human behavior. And that means that we have to start thinking in terms of responsibility at all levels of the organization, as Claire says, and we have to look beyond biology to how other organizations in complex adaptive environments manage their risks. We have to look at high reliability organizations and what they do in terms of creating the ability of everyone in the organization to speak up and to challenge anything which approaches an issue which might have high consequence. We have to organize where consequences are high to prevent those risks from happening.
So I'd like to follow up a little on that point, because I think this is a good point. I mean, I think even in our discussion here, it was clear that none of us who are supposed experts have written down exactly the solution to how this should be regulated. And I think the point that this is a really fast-moving field, which makes it even more challenging, is a really good point. So I don't know if anybody has any comments here. Like, what do you think we can do to create a dynamic environment which sort of promotes exchange and dialogue on these topics when it's very hard to write down a static set of rules which is going to stay current?
It's a difficult question. We heard about trust. We heard about things that are probably outside our academic world in industry, and regulations that are different in different countries. I think you could probably provide some guidelines as sort of a minimum, some advice. But we know from many fields, not only from biology or medicine, that regulating something globally is close to impossible.
Yeah, and I do think, to go to your question, the analogy that Volker just made with medicine maybe has some merit. I mean, the field of medicine is advancing very rapidly. There are regulations that grow up around it, but they're changing all the time. And I think figuring out how to apply that same sort of flexible thinking, recognizing risks and benefits, is an important question. Are there other questions in the field out there, sort of?
Very quickly, because I know we're getting out of time. I want to take issue with something I think you all agreed on at the beginning. Volker said that there is much more contact with wildlife these days. I don't think that's true. The rural population of the world is falling now as an absolute number as well as a percentage of the population. And even in rural areas, contact with bushmeat, wild meat, as opposed to farmed meat, is rapidly falling. So the vast majority of people are not getting their food from wildlife anymore or other contacts. And in terms of encroachment on wild habitats, again, the world is reforesting now, not in many places, but in a lot of other places it is, and in particularly southern China, there's been a rapid reforestation. So the amount, people are moving out of those rural areas into cities where they're having less contact with wildlife. And this was brought home to me when I had a conversation with an archaeologist who said, compared with our Neolithic ancestors who were obsessed with going into caves, we have way less contact with bats these days than we did then. By contrast, of course, lab risks are increasing because of the number of experiments and the amount of work going on. Just want to introduce a note of disagreement in that.
I think in this science of viral spillovers, essentially, you have two steps. The first one is the contact with the reservoir. The second is secondary transmission, human-to-human transmission. So even when you are reducing the first step, because you are right, there is maybe less contact, the increase in human density in all of those places increases the chances of a successful spillover as part of that two-step process.
Yeah, I would agree with some of what Gustavo said there. And I recognize Matt's point. I mean, from my perspective, an important aspect is, regardless of exactly which risks are going up and down, they're all real risks, right? They're all substantial risks, both wildlife, farmed animals, and biosafety incidents. They're all real risks. And, to some perspective, arguing which is a more real risk than another may miss the point that they're all real risks, right?
We didn't really. I don't know if we answered the question about pharma and organisations and their level of whether they have to report incidents, etc. That is quite an important thing because, of course, pharma are working in infectious disease areas very actively, and they're testing compounds. They're testing for evolution of resistant strains, and so we could regulate science in universities, let's say, but how does that feed through to organizations? And that's quite a big challenge, but it could be an opportunity, I think, of course, because university pharma partnerships are a big thing now. So, but I think it still needs to be on the agenda as part of any strategy. How do we capture work going in different areas?
I don't know who has the microphone, but I think there's a question over there.
My name is Jonathan Latham. My question is for Volker. Where do you draw the line between countries that obey the law in your terminology and countries that don't?
I cannot draw that line. I mean, that's exactly our problem. I can just tell what in my environment, so I'm working in Bern in Switzerland, so I have the feeling here everything is very well regulated, and I think that's probably in other countries, or not all other countries, it's regulated to the same extent. So for instance, we don't have institutional biosafety committees. We have it on a national level. So every application goes through the same channel. So these kind of things. But where you draw the line is difficult, because then you also go into politics, and it's hard to tell any country you're doing bad, and they will say, no, you're doing good. So it's very difficult to draw the line.
I mean, just one approach in terms of the building trust and kind of managing risk could be, for example, I mean, you have national clinical trials registries where if you're doing a trial, it's available on the web and you can read about it. You could envisage a situation where, to build trust, we submit a proposal online so that everyone can see the experiments you're doing, and the mitigations you have in place and the risk management plan, for example.
Yeah, I do think that's a good point. I wanted to quickly, since we're wrapping up, we just have a few more minutes. I wanted to go through the panel and sort of if anybody had any last comments they wanted to make. I know you were about to say something, Claire.
No, I was just going to say, I think it does differ between countries, and certainly, in the UK we have university level regulation and then national level regulation. But I know that what is containment level or BSL-3 in the UK is not necessarily BSL-3 in the US, for example. So I think we all agree that there are risks, but even amongst ourselves and the experts and the regulatory bodies, there are differences in assessing risk. So a good starting point might be to sort of unify that framework.
Well, a bigger challenge for me is the fact that depending who's funding your research, you need to follow certain regulations or not, which that opens a lot of big kind of worms.
Yeah, but that's what I mean. That's certainly different in different countries, because in some countries, including Switzerland, you don't need so the regulation or how you should conduct the work under which biosafety conditions is not dependent at all on funding.
Mm-hmm. I mean, something that Ravi mentioned, something that we've talked about before, is that, if you want to do animal work that is regulated, you have to justify in a grant application your numbers, why. And me as a reviewer, I would have to then comment on whether I think it's justified. You have to do the same thing with human work in clinical trials. There's no box on the form for a reviewer of a paper, and in many cases, to comment on the virology research. There's no box in many grant funders where you have to justify the experiments and the reviewers comment specifically. So just that sort of level of at least getting people being forced to think about what they're doing and people who are reviewing it think about might be a good first step.
Yeah, and this is a very interesting point that Clare makes, and I think it relates a little bit to what Ravi was referring to earlier in terms of sometimes using pseudoviruses. For those of you who aren't involved in research, you may know if you're doing both animal research and human subjects research, you're supposed to justify why you need to use those animals or why you need to do these experiments involving humans and why there isn't an alternative way to do it. And obviously in many, many cases, we do use animals or do experiments involving humans because there isn't an alternative. But for biosafety, there's currently no criteria that you justify why you're doing these experiments. And I do think this point that perhaps it could be thought about in that way is one at least worth considering.
I agree, and it's fairly straightforward. It's something you could action immediately. And we know that the funders do get together, and for example, for open access publishing, Wellcome Trust came together with HHMI and various, a few others, and maybe NIH, to make that happen. So there's no reason why they couldn't do the same thing for biosecurity in grant applications, especially with the amount of scrutiny that funders came under during COVID-19 for the work that was going on in Wuhan. So
Yeah.
I think that there is an impetus, and maybe they're already sort of planning it.
Anyway, I think we have about 22 seconds left according to the clock I'm looking at. So I'd like to thank all of you for coming to the panel. Before this began, I was often asked, like, how are we going to resolve all these questions? And I don't think we resolved them all, but I do really believe that, having scientists coming to these forums and really discussing these issues with people from diverse perspectives is an important step. So I'd really like to thank all of you for that. And we're now going to transition to the next panel, which is Global Biorisk Management.